Hi friends, in this short session we are going to discuss about ATCase, also known as aspartate transcarbamylase enzyme. Let's go ahead. Aspartate transcarbamylase is a cytosolic enzyme. That means it is present in the cytoplasm and it catalyzes the condensation of L-aspartate and Carbamoyl-phosphate to form another molecule called Carbamoyl-aspartate. Let's see how they look like. The reaction is this one. This is Carbamoyl-phosphate, here CONH2. Just a phosphate group is attached to the CONH2. It becomes Carbamoyl-phosphate, and this is L-aspartate amino acid. You know it very well. And in the presence of aspartate transcarbamylase enzyme, both of them they are condensed to give rise to a molecule called N-carbamoyl L-aspartate. And this is why this is important, means this is the first committed step in the pyrimidine biosynthetic pathway. What are pyrimidines? The CTP and UTP are the best examples for pyrimidines. So CTP and UTP are the two end products of this reaction. Which reaction? Here, from the N-carbamoyl aspartate, after several reactions, it lead to the formation of what? It lead to either CTP or UTP is formed from this after several reactions. And these are the end products of pyrimidine biosynthesis. This is the CTP molecule, you are well aware, and UTP, uridine triphosphate molecule. Why they are important means this CTP and UTP, they serve as cofactor in several reactions, and they also act as substrates in the RNA synthesis. RNA, you know that the RNA is made of adenine, ATP, GTP, CTP and UTP. They have to be synthesized as they are acting as raw materials for the synthesis of RNA. And also subsequent modifications of these CTP and UTP. For example, CTP, it gives rise to deoxy-CTP, and likewise whenever deoxy analogs are generated, they are used in the synthesis of DNA also. That is why these pyrimidine biosynthesis plays a major role in the synthesis of, in the formation of both the RNA as well as DNA molecule. Here, this enzyme ATCase or aspartate transcarbamylase enzyme, it is very important in regulating the formation of CTP and UTP. This aspartate transcarbamylase enzyme, it has two different kinds of polypeptide chains. One type is called catalytic polypeptide chains, another kind is regulatory polypeptide chains. They have two larger catalytic sites called homotrimers. Trimer means 3 units, and this is, we are talking about catalytic site, that is why it is called as C3, homotrimer, and 3 smaller regulatory homodimers. Homodimers means 2 units. Which one? This is the regulatory subunit. Is if it is represented by R, it is a homodimer, but catalytic is homotrimer. How it is, we will see. The catalytic subunits, they catalyze the carbamylation of the amino groups of aspartate. They catalyze the reaction. Simply they catalyze the reaction. Which catalyze the reaction? The catalytic subunits. See here, this C1, C2, C3, C4, C5, C6 are the catalytic subunits of aspartate transcarbamylase enzyme. They catalyze the reaction. Between what? Between the aspartate and Carbamoyl-phosphate. And their catalytic subunit, they consist of 3 chains. I told you C3 homotrimer, and these are not affected by the CTP. CTP will not come and bind to the catalytic subunits, remember. They just catalyze the reaction. Coming to the regulatory subunits, these green ones are regulatory subunits. They are also 6 in number. They contain 2 chains which are referred to as homodimer. That means regulatory subunit is in homodimer R2. and they do not show any catalytic activity. Catalytic activity is done by only catalytic subunits, C units, and these have, R units have regulatory function. They contain regulatory sites for the effector binding. What is effector? I told you in the previous video that effector is the molecule which is binding to the other site and affecting the enzyme activity. How it affects? It may either increase the activity or decrease the activity. Yes. ATCase holoenzyme, that means whole of the ATCase enzyme, the native enzyme, is made of 2 catalytic homotrimers and 3 regulatory dimers, making this a holoenzyme. This is the holoenzyme, which is dodecamer. Dodecamer means what? It contains total 12 subunits. And the native form of the enzyme contains 6 chains of each catalytic subunit and 6 chains of regulatory subunits with a molecular weight of around 310 kilodaltons. Remember, I told you this. It is a dodecamer, means 12 units, having 2 catalytic trimers and 3 regulatory. This is what it is saying, is this one. 2 catalytic trimers and 3 regulatory dimers, together making this as a holoenzyme. That means it is a multi-subunit complex. And each of the catalytic domain is composed of 2 structural domain. That means here each catalytic domain, C1, C2, each one, it is composed, it has 2 structural domains. One binds to the aspartate, another binds to the carbamoyl-aspartate, because the reaction has to happen between these two. That is why there are binding sites in the active site to bind both aspartic acid as well as carbamoyl-aspartate. And what the regulatory domain is doing? It is also composed of 2 domains. One binds to the, one is called allosteric domain, where the effectors are binding. Effectors, I told you there may be negative effectors or positive effectors, usually nucleotides. And another domain, the second domain, is the zinc domain. Okay, so the regulatory domain has, it again contains 2 slots, one to bind effectors, another to bind zinc. Why zinc is important? Means zinc is very necessary for the assembly of the aspartate transcarbamylase enzyme. That is why zinc, taking zinc is very, very important. Remember, the carbamoyl-aspartate and L-aspartate bind to the active site. Okay, the active site, where it is the catalytic subunits. Substrate binding, it induces, it activates conformational change. Okay, remember what I told you in the previous video about the homotropic effect. Homotropic effect means what? The substrate which altering the enzyme activity. Binding of substrates altering the enzyme activity. So binding of substrates, they increase the enzyme activity. How they are increasing? The enzyme, it changes from low activity, low affinity T state to a high activity, high affinity R state. What is called T state? I explained you that it is the tensed state. R state is the relaxed state where the enzyme is functioning its reaction, catalyzing its reaction. So there is a transition, interchange, continuous interchange between the tensed state to the relaxed state to the tensed state, and relaxed state is the one which catalyzes the reaction. In the tensed state, the reaction would not happen. Tensed state is binding to, is because of the binding of the negative allosteric effectors or inhibitors. Let's see, when the Carbamoyl-aspartate and L-aspartate are abundant, that means when the substrates are abundant, the enzyme remains in the R state. Okay, is it very clear now? When the substrates are available, they have to make the product. That is why the enzyme should be in the relaxed condition. Only in the relaxed condition it can perform the reaction. Once the substrates are exhausted, that means in the vicinity, if the substrates are not there, which substrates? Carbamoyl-aspartate and aspartic acid, if they are not present, the enzyme again changes into the T state. inactive state, we can say. Here there is interchange between the inactive state, this is the active state, and this is the inactive state. So by the binding of CTP, what happens? There is negative feedback effect, okay, which is nothing but the feedback effect means the molecule inhibiting its own reaction. Actually this is the reaction intended for the synthesis of CTP. If CTP is coming and binding to the enzyme, what it means? That CTP is present much in quantity, no need to synthesize further CTP. That is why it is inhibiting its own reaction, which is also called as feedback regulation. We'll see that later. But remember, whenever the substrates are exhausted, the enzyme returns to the T state. And let's see how the aspartate transcarbamylase enzyme is regulated. In the absence of substrate, I told you that the enzyme exists in the T state. When the substrates are available and when the substrates are binding to the enzyme, it shifts to the relaxed state and performs the reaction. Substrate binding to one site will increase the enzyme activity of the remaining sites. Did you understand? This property is called cooperativity, where binding of one substrate will activate or will increase the binding of more and more substrate molecule. That is called as cooperativity, which is seen in aspartate transcarbamylase. If the substrate is binding, then altering the activity, it is called a homotropic effect. What are the substrates here? Carbamoyl-aspartate and L-aspartate. They are responsible for the homotropic effects. And CTP and UTP can inhibit the enzyme. Which enzyme? Aspartate transcarbamylase enzyme, by weakening the R state. That means from the R state it is going to the tensed state. Why it is going to the tensed state? Or else why the binding of CTP or UTP inhibit the ATCase? Why means, if CTP is binding means the end product is binding. When the end product is binding, when the end product is synthesized in much quantity, it is going and binding the first reaction, saying that I am produced enough amount, there is no need to produce the same molecule. That is why it is going and inhibiting its own synthesis, called feedback mechanism. That is why CTP act as negative allosteric effector, or else we can say it acts as an inhibitor, CTP and UTP. Whereas ATP and GTP, they activate strengthening the R state. That means whenever ATP is present in the vicinity, they activate the enzyme. That means enzyme activity is increasing. It is converting more and more substrates to CTP. That means CTP production is increasing. Why it is increasing in the presence of ATP? Means one reason is ATP means high energy. As the energy is available, usually the synthetic pathways, they utilize the energy. That is why in the presence of ATP the reaction can complete. That means ATCase activity increases. Another reason is ATP means more purines. ATP and GTP indicates the presence of purines. What is this pathway? This pathway is the synthesis of pyrimidines. Okay, when more purines are there, remember the DNA structure. DNA contains equal quantity of ATP as well as GTP. When more purines are there, there is need for the synthesis of the pyrimidines. That is why for the second reason, if ATP or GTP are present, they indicates the much amount of the purines, thereby pyrimidines has to be synthesized. That is why the ATCase activity increases. These allosteric effectors shift the equilibrium between relaxed state and the tensed state. Which allosteric effectors? CTP and UTP, they act as negative allosteric effectors. ATP and GTP, they act as positive allosteric effectors or activators. Ultimately what happens? Due to this and maintaining the appropriate balance between the purines and pyrimidines within the cell. and this is the graph which is plotted between the velocity of the reaction and concentration, which is sigmoidal. This is the sigmoidal graph which is characteristic of allosteric. This indicates that ATCase is an allosteric enzyme. Clear? Thanks for watching.